Okay, so we'll continue our discussion about mycoplasma contamination in your cell culture. Normally, when you have animal cell culture that's growing in your bioreactor, you can get that contaminated by mycoplasma. Mostly the main source of mycoplasma contamination is from human people operating. So once that contamination happens, the animal cell culture will show some symptoms. Those symptoms are like, animal cells will become very sick because mycoplasma taking over the cell culture. They will, eat up most of nutrients. So, so animal cells will be depleted with the resources. So, they'll, biomolecular synthesis will stop. They will lose their nutrients, so they will change their morphology and they will sometimes die. Okay, you will see very slow cell growth and a lot of cells start die. So that's the typical symptom of mycoplasma contamination. So then if you see those kind of symptoms, you have to do some assay to make sure that indeed mycoplasma contamination is going on in your cell culture. Okay, so you cannot look at cell culture to see if there are mycoplasma there or not, because they don't smell like E. coli cells, or they don't change the cell culture turbidity. A lot of prokaryotic cells, when they grow, they will suspend around like E. coli cells. They will give you very turbid cell culture, and then they will smell as well. But mycoplasma on the other side, they don't smell like E. coli, and they don't become suspended, so they don't show you the turbidity change. Okay, so instead, the only clear way of confirming the mycoplasma contamination is by doing a real assay like ELISA assay or DNA fluorochrome staining assay, or biological culture assay, or biochemical methods of detection method, or PCR-based method. I think these days PCR-based assay method is very popular. So there are some kits that you can buy and test it. So if you confirm that yes, there is a mycoplasma contamination in your cell culture, what happens? The best thing to do is start it over. Just discard the cell culture that's contaminated and make sure you do clean it well and sterilize it well, so you don't get same mycoplasma contamination in your cell culture. Okay, so that's the best thing. So make sure you don't get any mycoplasma contamination in your cell culture. Okay. And then we're talking about different types of filtration. We talked about microfiltration. So microfiltration cannot remove mycoplasma, of course. So they will go right through. But ultrafilter may be able to remove mycoplasma if you decide to use it. Okay. Anyway, these are the filters that are with very small pores. So they can remove viruses, or they can remove, endotoxins, mycoplasma, all those things. Okay, so ultrafilters are used. Among the usage of ultrafilters, yes, there's something called reverse osmosis that will use ultrafilters. So reverse osmosis, the name comes from the description of water molecule flow direction. Okay, so osmosis is what? It's a movement of water molecule based on their concentration gradient. Okay, so diffusion is the concentration gradient based molecular movement, right? So water diffusion is basically osmosis. So if you have a lot of water will travel from higher concentration to lower concentration. That's what osmosis is. But when you say reverse osmosis, what does that mean? What that means is water molecule will move opposite to osmosis direction. That's because there's going to be a huge pump that's going to pressurize water, so the water will move against their concentration gradient. That's what osmosis is. So if there's a semi-permeable membrane, water molecules can go through that membrane. Then yeah, you press water, so apply pressure, so water molecules will move through the membrane. But that membrane is not permeable for some of the contaminants, so they will be captured. So that's what reverse osmosis is. Normally that membrane is ultrafilter membrane, so it is used as a water purification system. Okay, and filtration system. So we talked about different size filters, right? And then how do you filter out your contaminants? So you can use small scale lab filtration system. Depends on how much of the feed material you're gonna filter out. And then you can use maybe vacuum pump, or you can use a syringe using your maybe finger pressure. You can run your feeding material over the filter, or you can sometimes use centrifugation force to move the feed over the filter membrane. Okay, if you have very small scale filtrate to run through, you can use these different methods. Okay, and you can look at the table figure in your textbook to understand what those are. Syringe filters are used a lot for these types of small scale filtration. And then large scale filtration, just like biomanufacturing operation, like that, they may have 100 liters or 1,000 liters to filter through. Then there's no way you can use any of these systems. They'll be so inefficient, right? Then you have to use a large pump that can handle the large size filtrate, of course. Okay, and during your large scale operation, there's a process called clarification. That happens between the upstream process and downstream process. So through this step, you can separate out soluble proteins from cells and cell debris that you don't want. Okay, so filtration is preferred over centrifugation for this step because it is less expensive and it's gentler. Centrifugal force is greater. Sometimes you can smash the molecules, and it is more convenient. Okay, so clarification is a large-scale filtration method. Okay, and depends on how you run your filtrate over your membrane. Okay, so the direction of the flow over filter system kind of matters, because if you try to filter your initial material over your filter membrane kind of vertically, okay, we call that as a normal flow filtration or direct flow filtration. Okay, it's just like your coffee filter. Okay, so if you pour in your initial material, yes, the things that will filter through will go through. It'll be captured vertically at the bottom, right? So sometimes you can shake it up to have more things that can go through. But there's a problem. The problem is, as your coffee filter you use in the morning, you get clogged the filter very quickly. Okay, so if you look at the filter rate, flux rate, and volume filtered, okay, now as soon as you filter some volume, the flow rate will stop, because you have a lot of these fouling effect. The fouling is blocking the passageway, so you have dead end filtration. The filtration is stopping if you use this direct or normal flow filtration. Okay, so normal flow, it's fine if you have very small volume to filter through, but if you have a large volume to filter and remove, and this is not the method you want to use, because it presents the dead end fouling effect in your filtration process. So normal flow filtration is vertical flow of your initial material through your filter. Okay, then is there an alternative? Yes, of course. The alternative method is something called tangential flow filtration or cross flow filtration. Okay, the way it works is, if you have membrane there or there, okay, and instead of running your feeding material over the membrane directly, you will move them kind of sideways, kind of parallel to the membrane surface. So the initial material will continuously move parallel to your filter surface. So since the main force will sweep them away, there will be much less clogging and they will be washed away. So there will be continuous filtration happening, although it may take a long time to filter most of the material. So tangential or flow filtration would not present you with the clogging or dead end problem. So yes, it's going to continue its filtration for a long time without you have to replace the filter. Okay, so if you are doing a large scale filtration, yes, definitely tangential flow filtration is the method you want to use. Okay, so that tangential flow filtration, another name for that is a TFF method. TFF, you will hear a lot. Tangential flow filtration, TFF. Okay, feeding material going tangential to the filter surface, going parallel to the filter surface. The benefit of doing that is no internal plugging, okay? Low pressure resistance, high flux performance. These are all the benefits of using tangential flow filtration. So if you look at the tangential flow filtration filter design, so it looks like this. There's a filter there inside a cassette, and then main flow will take place parallel to it. Okay, so the initial material comes in, it's going to go sideways, it goes out. Okay, you're going to circulate over and over, and there are some residual filtration happening. So you will capture, you'll permeate what's filtered through your membrane from the sideway. Okay. So tangential flow filtration reduces the clogging problem since the fluid is filtered through over the surface of the filter as well as through the filter. Okay, so the main flow will sweep away any kind of dead-end clogging or filter cake that's sitting on the filter surface. So it's going to continue filtration for a long time. So TFF is used a lot in the biomanufacturing industry. It is applied in these main steps. Number one, clarification step. We talked about clarification, right? Removing large cells and debris between the upstream and downstream process. Yes, TFF is used during this step. And purification. Yes, you can purify out small molecules from large molecules. Yes, TFF will be used for that. Concentration. Concentration, I told you, it's a way to concentrate your starting material. So concentrate means what? You selectively remove the solvent only without losing your solute or molecule that's dissolved in the solvent. Okay, by doing so you can bring up the concentration. And then diafiltration or buffer exchange, they are interchangeable words. Diafiltration, buffer exchange, what that means is you can change the buffer, okay, that has your API, for example. So let's say your API is dissolved in a tris buffer, okay, but tris buffer is not comparable for your next process of your purification. So let's say it's not comparable for an exchange chromatography or something. So you have to change the buffer. Okay, and how do you do that? Yes, you can use TFF for that buffer exchange process. Okay, you can remove the old buffer by removing your solvent only, and then at the end the concentrated API will be reflushed in a new buffer. So that's what diafiltration or buffer exchange step is. So TFF can be used for these four main applications. So TFF is used a lot in the industry. That's the key concept. So TFF system can come in a small scale tabletop model or large scale flow top model. So yes, when you purify your API in a large scale, yes, you use TFF system like this. Okay, and then whatever the system you are running, this is kind of simplified diagram showing the system run of your TFF. So there's the TFF cassette, membrane is in there. So your initial material comes in from the initial tank through the pump. It comes in, and your retentate will go out, and it will be recirculated over and over, and the permeate will come out of this way. Okay, so that's will be the simple makeup of your TFF system. And then so the steps in the TFF operation, you will probably start with the flush. The entire system will be washed with OFI. Okay, and then CIP, clean in place. So before you use the TFF unit, you have to clean up the entire system, the buffer tank, and then connecting tubes, pump, and then TFF cassette, and everything should be washed with a solution like sodium hydroxide, one normal solution. We'll get rid of those, and then you do the flush with UFI again to remove the residual amount of sodium hydroxide, and then you're going to do the conditioning. Conditioning means you treat the entire system with the buffer that you will be using. So that's the conditioning step. And then you will do your processing, whatever the process is, maybe concentration operation or diafiltration operation or purification, you do that. And then at the end, you recover your product, and then you clean in place again using sodium hydroxide solution. And then you do the testing. I will talk about the testing in a second.